So today's topic is an introduction to DNA, and I've taken some notes from the lesson microbial genetics, and we'll be seeing it. Okay. See, microbial genetics provides powerful tools for studying the regulation as well as the function pathway of the organization of cellular processes. It plays a major study of microbes and significant role in development of molecular biology. So let's get into the lesson. Okay, what is a DNA? So DNA, the full form of DNA is deoxyribonucleic acid, and it is a polymer composed of two polynucleotide chains that coil around each other to form a double helix carrying genetic instructions for the development, functioning, growth and reproduction of all known organisms and many viruses. So if we see, the DNA is present in all the organisms including viruses, and it has two polynucleotide chains. Okay, they coil around each other and form a double helix. Okay, they carry all genetic instructions and they play major role in development, function, growth and reproduction. DNA and ribonucleic acid are nucleic acids. DNA is made up of chemical building blocks called nucleotides. Actually, they are small particles and they are called as nucleotides. It stores instructions for making other large molecules called proteins. These instructions are stored inside each of your cells, distributed among 46 long structures called chromosomes. These chromosomes are made up of thousands of shorter segments called DNA called genes. So gene, in simple words, gene is defined like it is a unit of heredity which is transferred from a parent to a progeny or a child. The DNA is transferred, or the heredity or the information is transferred from the parent to the progeny. Okay, so this is the shorter form for gene. So it's a very simple term, so that you can use for the definition of term genes. Okay, this is the structure of DNA. So you can see here, it forms a helix structure and it has a base pair actually. So if we see here, see the adenine, it pairs with thymine. Okay, so A pairs with T and guanine pairs with cytosine, that is G pairs with C. So if we see adenine is given in yellow color, so it pairs with thymine, that is T. Okay, and guanine, which is red, pairs with cytosine, which is in orange color. Okay, this is a basic rule. Okay, we'll be seeing it the coming slides. Okay, so what is nucleotide and nucleoside? Okay, each nucleotide is made up of three components, namely, so the three components are nitrogenous base, sugar and a phosphate group. Okay, so nucleoside is nothing but it's the nucleotide without a phosphate group. That is, the nitrogen base and the sugar are only present and the phosphate group is not present. Nucleotide without a phosphate group is known as nucleoside. The sugar present in the DNA is deoxyribose sugar. So deoxyribose sugar is a component of pentose sugar. So pentose sugar is of two types actually, deoxyribose and ribose sugar. Okay, and the deoxyribose sugar, we'll be seeing the structure, and that's all. So it's a type of pentose sugar, and there is also a ribose sugar present, and we'll be seeing that in the next videos coming. Okay, the nitrogen bases present in DNA are actually two. Okay, they are purines and pyridamines, and the purine, it's nothing, the adenine and guanine. So adenine is represented by A and guanine is represented by G, and pyridamines consist of thymine and cytosine. The thymine is represented as T and cytosine is represented as C. The nucleotide is a unit formed by the glycoside bond between nitrogenous base and sugar ester bond between phosphate group and sugar. Each of the nucleotide is bonded by a phosphodiester bond to form a polynucleotide chain. Okay, so each nucleotide is bonded by a phosphodiester linkage. Okay, they form a polynucleotide chain. Okay, there are many enzymes involved in DNA replication. So the DNA replication is nothing, but it includes like there are major steps actually involved in the replication. There are three types of replication. There are possible ways. It's semi-conservative, conservative, and dispersive. Okay, it's the three DNA replication possible. And if we see, okay, the enzymes involved in the DNA. So we'll first see helicase. Actually, it unwinds the DNA. So the two polynucleotide forms the helix. Actually, it unwinds it. Okay, and the next one is DNA gyrase. It relieves stress created by the unwinding. So when the DNA is actually when it's unwinded, there is a topological stress caused, okay, in the DNA. So it relieves the stress, and SSB protein, that is single stranded protein. Okay, single stranded protein. There is single stranded binding protein. Okay, it binds to single stranded DNA and stabilizes it. And primase, it is enzyme involved in the synthesis of RNA primer. Okay, RNA primer is an important unit in the DNA. And DNA polymerase 1, it plays important role in excision of primer, and DNA pol 2 is there, but it is a very very slow process, and I have not included this. Okay, included the DNA pol 2 here. And DNA polymerase 3, new strand elongation, DNA ligase, it joins the nick. Okay, there are many laws and theories in DNA, and for our class there are very less laws and theories. Okay, first we'll be seeing the Frederick Griffith experiment. Okay, yes. Okay, Frederick Griffith experiment. Okay, in 1928, British bacteriologist Frederick Griffith was trying to develop a vaccine against pneumonia. In his experiments, Griffith used related strains of Streptococcus pneumoniae. Okay, he used two strains, that is rough strain and a smooth strain. Okay, the rough strain is a virulent non-capsulated strain. It forms rough colonies on the culture media, and the smooth strain is virulent capsulated strain forming smooth colonies on the culture media. Okay, Griffith injected live smooth strain into mice, which caused disease and killed it. When he injected live rough strain into mice, it did not cause any disease, and mice remained live. Okay, so but the experiment gives surprising results when a mixture of harmless live rough strain and heat kill smooth strain was injected into mice. Griffith observed that the mice developed pneumonia and died. Further, he analyzed the blood sample from the dead mouse. He found that it contained live smooth strain. This accidental discovery made Griffith to conclude that the rough strain changed into smooth strain by taking up a substance which he called transforming principle. Okay, so this is the one of the important experiment and theory. Okay, Erwin-Chagas rule. Actually, this is a very very important rule, and it's actually related to the DNA. Okay, so if we know this rule, we'll be able to know the structure also. Okay, it's some related to it. Okay, Erwin-Chagas measured the quantity of bases in DNA and noticed that the number of adenine, that is A, is equal to the number of thymine, that is T, and the number of guanine, G, is equal to the number of cytosine, that is C. So if we see here, the adenine pairs with thymine by double bond, and the guanine pairs with cytosine by triple bond. Okay, it is a very important note, so you can just make a note if you have some space. Okay, so this is a measurable quantity. Okay, A is equal to T, or A divided by T is equal to 1, or C is equal to G, or C by G is equal to 1. Okay, so A plus G is equal to T plus C. Okay, it comes. Okay, so if we see here, yeah, so adenine, it pairs with thymine, so we saw, right? So adenine pairs with thymine and guanine pairs with cytosine. So adenine pairs with thymine by double bond and guanine pairs with cytosine by triple bond. Okay, this is an important note. Adenine pairs with thymine by double bond and guanine pairs with cytosine by triple bond. Okay. So here we have abbreviations and important terms. Okay, so see, SSBP, that is single stranded binding protein, and DNA, yeah, we saw it, right? Deoxyribonucleic acid, gyrase, actually it releases a topological stress produced by helicase actually. Okay, so gyrase, it is an enzyme, it produces topological stress produced by helicase. RNA primer, short strand of RNA that serves as a starting point of RNA synthesis. Okay, this RNA primer plays a major role in RNA synthesis. So bacterial genome size. So bacterial genomes is usually represented in MB. Okay, is represented in MB, and if we see here, which is the genome which is represented by MB, the gene density is high, and if it is tiny, it is represented as KB. Okay, it is also high, and in if it is large, it is represented as GB, which is low. And if we see example for MB small, Escherichia coli has 5000 genes, and if we see tiny, KB, that is in virus and organelles. Bacteriophages has 10 to 100 genes, and in eukaryotes large GB is seen. Okay, it is actually gene density is low, and Homo sapiens to 25,000 genes. Okay, it has 25,000 genes actually. Okay, and the questions for better understanding here. See, the Irwin-Chargaff rule is a very important question, and it's very simple. Adenine pairs with thymine by double bond, and guanine pairs with cytosine by triple bond. Okay, that's okay. And draw the structure of deoxyribose. This is a many time asked question actually. So I told you, right, the deoxyribose is nothing, it is a type of pentose sugar. Pentose sugar is of two types, that is deoxyribose sugar and ribose sugar. Okay. So if we see here, see the pentose sugar, actually the chemical formula I will be writing. So C5H10, this is H, okay, H10 and okay, this is 0, okay, 0,4, okay, that's all. And okay, see, so pentose, right? So pentose sugar, so pent means, we know that pent means 5. Okay, pent means 5, so you will be having 5 carbons. Okay, you will be having 5 carbons. Okay, so see, first we have an oxygen atom here. Okay, so and next it's a pentose, and it will be having 5 carbons. Okay, it's like the shape of a pentagon. Okay, that's all, it's a shape of a pentagon, and we'll be having C. Okay, this is carbon. So if we see, here is 1, 2, 3 and 4. Okay, 4 carbon is present. This is 1, 2, 3 and 4, and we'll be attaching here also HO, CH, HOCH2. Okay, Okay, and here OCH2. Okay, and here see, here we have OH and H, and again we'll be having OH H. Okay, so here H will come. Okay, and yeah, here also H will be coming, so it's here oxygen is not possible, so H. Okay, and here also you'll be having an H. Okay, here also you'll be having HOCH. That is, this is the fifth one. Okay, this is the fifth one, that is spent 1 2 3 4 and 5. This is 1 2 3 and 4, this one is 5. Okay, it's a very simple diagram, and this is a type of pentose sugar. You can just, oh, it's very easy diagram. Okay, and what are three general methods of replication? So three general types of replication actually we saw, right? It's semi-conservative, conservative and dispersive. So what is semi-conservative? DNA replication, it produces two copies of double-stranded DNA, that is dsDNA, each containing one old strand and one new strand. In conservative, DNA replication produces two daughter dsDNAs, one of which consists original strand and the other one consists the newly synthesized strand. Dispersive, DNA replication which original dsDNA undergoes fragmentation. The fragments synthesize the structure of which it formed the two replicas. Okay, and it is also the three replication methods. Okay, the first one is called as theta mode, and the second one is called as sigma mode, and the third one is called as linear mode. Okay, these are three type of replication. It is seen in eukaryotic organism and many viruses, okay, which consist of the linear DNA molecules. Okay, the chromosomes of eukaryotic organisms and the viruses which contain linear DNA molecules. It undergoes the three major replication, that is theta molecule, sigma molecule and linear molecule. Theta mode. Okay, that is theta mode. Okay, this is theta mode. Sigma mode, and the last one is linear mode. Okay, and okay, this is high order thinking skill question. Okay, and it's a very important question, and it's like we'll have to have concentration here. Okay, if a percentage of adenine in the DNA strand is 20, so understand the question. Adenine in the DNA strand is 20. So the Irwin-Chargaff rule says that adenine pairs with thymine. That is the number of adenine is equal to the number of thymine. That is A is equal to T. Is equal to T by double bond. Okay. So if the adenine of one strand is 20, so adenine is 20, then thymine is also 20. Okay. Can you determine the percentage of other bases? So what are the other bases? Only 4 nitrogen bases are there. That is adenine, thymine, guanine and cytosine. Okay, and the adenine is given as 20. So thymine is also 20. So 20 plus 20 is equal to 40. And so the other, see, if in 100, if we minus 40 means the remaining is 60. Okay, and 60 if we divide by 2, that is for cytosine and guanine, if we divide by 2, it is 30. Okay, So adenine is equal to 20%, thymine is equal to 20%, and the remaining 60%, if we divide it by 2, that is for guanine and cytosine, it will be 30%, and guanine is equal to 30% and adenine is equal to 30%. Okay. So A is 20. Okay. So A is equal to T, that is adenine is equal to thymine. So T will also be 20%. Okay. And if we add this, it will be 40%. Okay. So 100% is there. So if we minus 100 minus 40, it's 60. So 60% remaining. Okay. So 60%. So 60%. The 60% is nothing. It is composed of adenine. Guanine and cytosine is together as 60. So guanine and cytosine, if we divide by 2, okay, that is divided by 2, okay, it gives 30%. So adenine is equal to 20%, thymine is equal to 20%, and cytosine is equal to 30%, and guanine is equal to 30%. And if we add all this, so 20 plus 20 40, and 30 plus 30 60, together is 100%. Okay, I think the concept will be clear. Okay, so what is a gene? So gene is a very simple term. If you want to use other term, you can use, but a simple term, okay, to learn what is gene is, it is a unit of heredity. Okay, gene is a unit or heredity, okay, which is transferred from the parent to the progeny. Progeny is the it's from the parent, okay, from the parent to the kid, the parent to the son, or from the parent to the daughter. Okay, that's progeny. And the progeny, it will be transferred from the daughter, it will be transformed to the granddaughter, from the granddaughter to the grandson. So it will be like that. Okay, it's a very easy lesson. Okay, so thanks for watching, and we'll be learning about the alternative forms of DNA and the helical sizes, sense and diameter in the coming video. Thanks for watching.